Hey everybody, this is going to be the summative 7C learning target review. I would encourage you that if you don't understand how to do dihybrid crosses or especially monohybrid crosses, you go back and you watch those videos. This first video, I go over all of the terms that we're going to be using interchangeably in here. So if what I start talking about doesn't make any sense, go back and watch those other two videos so you can become familiar with the vocabulary. The 7C learning target says I can apply non-Mendelian inheritance using Punnett squares and pedigrees to predict possible outcomes of various genetic combinations. And in order to be able to do that, I need to understand how sex-linked traits are passed from parents to offspring. I need to be able to use Punnett squares again to look at codominance, multiple alleles, and incomplete dominance. And I also need to be able to look at pedigrees and identify the genotypes of individuals, figuring out is it a dominant gene or recessive gene, is it autosomal, or is it sex-linked. So the first thing we're going to do is we're going to go over 7.7, these codominance, incomplete, and multiple allele terms. Now I have linked this review chart into Canvas, so you feel free to look at it when you need to. And all it has is all of the terms that we've talked about in this unit, divided by simple inheritance and complex inheritance. So in this video, we're going over complex inheritance or non-Mendelian. So in simple inheritance, it's just one dominant, one recessive, the end. Really easy. But unfortunately, not everything in our body is that simple. So one of the first terms we needed to know was polygenic traits, and that's when a phenotype is influenced by more than one gene. So the example that you're given in the PowerPoint is skin color and height. We're not just one shade or another, we're a bunch of shades in between. And that's because it's influenced by more than just one gene. So in this example, we have 3 genes that influence whatever characteristic we're talking about. Incomplete dominance is when we have 2 genotypes that blend together to make a third phenotype. So instead of red flowers and white flowers only, we also have pink flowers. Codominance is when we have 2 alleles that are both represented in the phenotype. So instead of only red flowers and only white flowers, we have red flowers with white polka dots or white flowers with red polka dots. They both show up. They're not blended together like incomplete dominance. Multiple alleles is when we have more than 2 alleles for a specific gene. So the example that you're given in the problem set is blood types. You have, instead of just big A and little a, you have big A, big B, AB, which are codominant, and then the recessive is O. And lastly, for sex-linked, it's when a trait is influenced by a gene found on the sex chromosome. So when you think about karyotypes, remember the little pictures of the X's, that last karyotype is your sex chromosome, your 23rd pair. And if there's a gene found on there that we're talking about that's passed down, that is a sex-linked chromosome. So it has to be found on either the X or the Y, and normally it's found on the X. A word that you need to be familiar with and associate with sex-linked characteristics is being a carrier. So a carrier is an individual who has the gene for a specific disease or trait, but is not affected by it. They're able to pass it on to their offspring, and their offspring might have it, but they do not express the disease or trait in their phenotype. So let's look at some of the practice that we were supposed to do for X-linked traits. So for this first one, we have a disease called Becker's muscular dystrophy. Is a rare recessive disease typically found in males in which the muscles atrophy, meaning they break down over time. Becker's is an X-linked trait. So this bit of information right here tells me that this disease is found on the X chromosome because it's an X-linked trait. Fill in the table to complete the genotypes for each individual described. It also says in this problem that it's a rare recessive disease. These two bits of information are extremely important, and they're two things that you need to look for when you're doing X-linked problems. Because we need to know, is it a big letter or a little letter? This piece of information tells us it's recessive, so we're going to be using a little letter. And then we also need to know, do we put it on the X or the Y chromosomes? Remember, there's two sex chromosomes. This tells us it's an X-linked, so we know that it's going to be a little letter on the X-linked. And if it's recessive, the only way a person can be affected by muscular dystrophy is if they have two of the recessive alleles, or in males, if they only have the recessive allele. All right, so we need to figure out the females and the males' genotypes. The first thing we should do is we already know the codes for females and males. So females are XX and males are XY. So I'm going to go ahead and put those in there already. XY for a male, XY for another male, XX for a female, and XX for a female. Okay, so I have their sex chromosomes listed, and now I need to figure out, based on what it tells me in the individual phenotype, where to put these recessive alleles. And because this is Becker's muscular dystrophy, I'm just going to use a B. You can use whatever letter. I'm just going to use a B because the name of it starts with a B. A female carrier. So if you remember from our genetics cheat sheet, a carrier is somebody who has one of each. They're heterozygous. So this female carrier, being heterozygous, knowing it's recessive, that means she has the recessive allele, but she's not affected by it. So that means she also has the dominant allele attached to the other X chromosome. So a carrier is somebody who has both. They have that recessive allele, but they also have the dominant allele, which takes away the disease that would be shown up in the phenotype. A male without Becker's. So again, this is a recessive disease. So if this person had it, this male would have a little b, but instead they do not have the disease. So we're going to put a capital B in there. A male with Becker's syndrome is going to have that recessive allele. So on their X chromosome, because it is X-linked, we're going to give them a B. A female with Becker's syndrome is Becker's muscular dystrophy is going to have two little b's because it is a recessive disease. And in order to have muscular dystrophy and a female, you have to have two lowercase b's or two recessive alleles. And then lastly, a normal female is going to be two capital B's. So this person isn't even a carrier. They can't pass it on to the next generation because they don't have it. So that's how I would work out the genotypes. If you're still confused on this, rewind the video and go back. All right, number 2. A disease known as XLA or X-linked A-gamma globulinema is an X-linked recessive genetic disorder in which individuals that have the disease do not make certain white blood cells in order to properly fight infection. For each genotype listed, describe the individual. Okay, so the first two things I need to know, is it on the X or the Y chromosome, and then is it dominant or recessive? I know right here it's X-linked, so I'm going to highlight that. And it's a recessive genetic disorder. So back to back, I get the information that I need. It's X-linked and it's recessive. So in order to have it, it needs to be either two of the recessive alleles on a female, or a male needs to just have this recessive allele, the lowercase. So this individual first, let's go through and figure out if they're males or females. So this is an XY, so it's going to be a male. Here we have XY again. Here we have XX, so female, female, and female. All right, this is a recessive genetic disorder, so I'm looking for lowercase a's. This male is going to be normal because they have a dominant allele. This male is going to be with the disease, XLA, because they only have the recessive. This female is going to be normal because she has no recessive genes. This female has one recessive and one dominant. So, is she affected by the disease? No. But does she still have that gene? Yes. So this female is a carrier of XLA. And then this female with two lowercase a's is with the disease, XLA. All right, now let's take these genotypes and let's do a Punnett square with them. A woman who is a carrier for XLA marries a man who does not have the disorder. They have 4 sons. How many could be expected to have XLA? So we have a female who is a carrier. So I'm going to take this genotype here and I'm going to separate the alleles. Into my 2 boxes with a male who does not have the disorder, so a normal male. I'm going to take the 2 alleles here. Okay, and now I'm going to cross them. So in this first box, I'm going to take this XA big A and this X big A and combine them. Here I've got X a little a and X a big A. I'm going to put the big A first just out of formality. And now we are going to do the Y's. So I have the X with the dominant A and a Y. And in this one, I have X recessive A and a Y. Okay, so I filled this out. The question was, they have 4 sons. How many could be expected to have XLA? It's really important that you look at what they're asking, because here they're asking for males, not females. So I'm going to be looking at this bottom row here, not the top row. So if a woman who is a carrier and a man who does not have the disease have 4 sons, how many could they expect to have this? Well, if I look down here, there's a 50% chance that their offspring are going to have that disease, because the bottom row here is going to be the genotypes for the males. They can either have a male who does not have it or a male who does, who got it from mom. So if they have 4 sons, we could expect that 50% or 2 out of the 4 are going to have XLA disease.